Hi, my name is Nan McDonald, and I am Chief of Data Operations for the Institute for AI in Medicine. This is the fourth of a series that's being brought to you by the Center for Collaborative AI in Medicine. I will turn it over to the speakers. If you have any questions, please use the Q&A. And if you have any suggestions for future topics, please email us and let us know. We really thank the students that have joined today. We're really excited to hear what they've discovered. And so without much further ado, I will turn it over to Megan.
Yes, so good morning, everyone, and thank you, Nan, for the introduction. I hope everyone is staying nice and warm today with the snow. And so I'm excited to get started. The next slide would be great. Thank you. And so I'm excited to introduce this paper that was recently published in NPJ Digital Medicine this past summer called Mining the Contribution of Intensive Care Clinical Course to Outcome After Traumatic Brain Injury. And so to provide some background, I think the authors really aptly describe traumatic brain injuries as a progressive condition rather than a single event. And I think this captures the very complicated nature of these types of injuries. And so, for example, after neurotrauma, patients are at an increasingly higher risk for secondary injuries that result after that initial trauma. So, for example, after the initial trauma, there can be blood in the brain, and this can start to expand over time within the preceding few hours. You can imagine this can be quite catastrophic. It complicates a patient's clinical course. They may be in the hospital for longer and be at a higher risk for long-term disability or even death. And because of the heterogeneous and evolving nature of TBI, it's been difficult to derive individualized treatment approaches for these patients. But one of the benefits of the intensive care unit, at least from a data science and analytics perspective, is that it's a very data-rich environment. And so patients with traumatic brain injuries may be hospitalized for days or weeks, even longer. And over the course of their admission, there's a lot of data that is generated and collected. And so patients may be on multiple medications where the dosages and even the medication types are changing. They have repeated monitoring of their vital signs like heart rate and blood pressure. They have repeated imaging. They may undergo surgery or other forms of neuromonitoring, and they have frequent laboratory tests. And so this creates quite a wealth of data that the authors will leverage for their advantage, as we'll see. And so their primary objective for the study was to develop a modeling strategy that can better delineate the clinical trajectories of patients with TBI. And so there's three sort of main sub-aims here. So one is to leverage all of the electronic health record data that is available to predict a patient's 6-month functional outcome after their traumatic brain injury. Second, they attempt to quantify the added benefit of having models that have both static and dynamic data, where static data is those variables that don't change. So a patient's sex or race, ethnicity, and then dynamic data are things like blood pressure or heart rate, which are changing frequently over the course of a patient's admission. And lastly, they also develop a method to identify clinical events that are most strongly associated with transitions in an individual's clinical trajectory. And in the long term, the overall objective here would to help facilitate precision medicine efforts for traumatic brain injury patients. And so they use the Center TBI dataset. This is a large prospective observational cohort study of patients, all whom have traumatic brain injuries from across 65 centers in Europe. And it contains almost 1,200 different variables, both static and dynamic. And it also includes 6-month outcome data in the form of Glasgow Outcome Scale Extended, or GOSE scores. And this is an ordinal 8-point scale that's used to rank functional outcome at 6 months post the traumatic injury, where a GOSE of 1 indicates that the patient had died by 6 months, and a GOSE of 8 at the top of the scale indicates that they had the best possible recovery. And so getting into their modeling strategy, I think they use a really interesting approach that I've started seeing similar adaptations elsewhere. And so, as we mentioned, over the course of an ICU stay, there's a lot of data that's being collected. And in order to integrate this data and be able to learn from it, they essentially use this tokenization approach. And it's similar to if we think about natural language processing, where they're taking variables and they're kind of formatting them into words or tokens. And so, for example, if we look at the left, for variables such as age, or continuous variables rather, what they do is they take the name of the variable and then they append it the quantile bins. So for example, they discretize continuous values and they find the associated quantile bins. So for example, this patient looks to be 46. And so this corresponding data point would then just be represented as age underscore bin 8. And for categorical variables, they just take the name of the variable like sex, and then they append to it that value. So sex underscore M or F. And they do this for all the different categorical or continuous variables. And also notably, they also, instead of imputing missing values, they represent the missingness by appending NA. And this is so that they can help identify whether there are patterns in the missing variables that could be informative for predicting a patient's outcome. And then after that tokenization process, so all the variables are tokenized, they perform this embedding and weighted aggregation step. They're a little stingy on the details here, but from what I understand is for each token, they trained a model to predict the 6-month GOSE outcome. And they then took the corresponding embedding and they replaced the token with that embedding. And so this is a way of integrating and taking into account all the information from the other tokens within all the other information as tokens within a certain time window, and just representing that token with some more kind of granular, broader information. And so for each time window, they then take all of the corresponding tokens or the embeddings for those tokens, and they average them, and they also weigh them by this relevance weight that they learned while training the model. And so what happens after this kind of complex process is that they have this one lower dimensional vector for each time window. And so this lower dimensional vector is then fed into a recurrent neural network. And so essentially what happens is that at each time point, the RNN takes information from that, it's a 2-hour window, and that it provides a prediction of a patient's 6-month outcome. And it provides those predictions in the form of thresholds of the probability of the outcome for each GOSE. So here in the blue, we have a GOSE greater than 1. So essentially the patient is going to at least survive. And we can see what these probabilities are at each 2-hour interval. And so in this way, the model is able to learn from subsequent data over time and constantly update its predictions. So it's a very dynamic modeling process. And then importantly, what we'll see in the results is that they compared their models with what's called the impact extended score. And this was a previously validated model from 2008 that was also designed to prognosticate 6-month functional outcomes after TBI. And what's notable here is that it's calculated with only 10 variables that are recorded within the first 24 hours of admission. And this is one of the kind of mainstay models that we see in a lot of papers. So this is widely used in practice. And then lastly, another component was to identify high magnitude clinical transitions. And so we can see, or I see Eva, your hand is raised. Should I stop?
Sure. I think one of our participants has raised their hand. So if there's if there is a question, if you could please type it in the Q&A, that would be probably the best way, and then we'll share those out.
All right, thank you. Yes, please. And so, so this next section, they were also have an approach for identifying what they call as high magnitude clinical transitions. So for this, they use a previously developed but still recent algorithm called TimeShap. And this is an interesting technique which allows us to kind of add some interpretability to the neural network predictions. So it's able to quantify which features or variables were most influential in a neural network's prediction. And so in this case, TimeShep specifically was applied to identify which clinical variables or events in the hospital were associated with the transition in a patient's clinical trajectory. So for example, which medications were associated with a patient worsening or improving. And we'll see some examples with that. And then next slide, please. Okay, thank you. And so getting into the results, this first slide is showing the results of their full model. So here in the blue is their full model with the almost 1,200 different variables. And they're plotting how much of the variance in outcome was identified with their model. And then they're plotting it over the days since ICU admission. And so we can see kind of at the highest point, using the full set of variables explained about 51% of the variance in a patient's functional outcome. And we can then look here in the red, this line is showing us the explanation of variance for the just the impact model with those 10 variables within those first 24 hours. And we can see that, you know, maybe at its highest point, it looks about 44% of the variance. And we can see, you know, with both of them, they are decreasing over time for the full model. After about 3 days of the ICU, it does start to decline in how prognostic this model is. But then what's also we can see here on the bottom panel is the number of days before ICU discharge. And so within the last week, that explanation of variance starts to increase for both models. And so a few takeaways here is that I think one, with you know, even with the, you know, over 1,000 extra variables for this full set, you know, we don't see that substantial of a difference in how much variance is explained between the two models. I would have, you know, intuitively maybe thought there'd be a lot larger of a difference. And then it also, these results also suggest that the first 3 days of ICU admission and the last 3 days or the last few days before ICU discharge are explaining a lot more of the variance. And so these days in particular may carry more prognostic weight. And then next they looked at the added explanation of variance with their full model. So here in red, this is showing the added explanation of variance of the full model with 1,200 variables compared to their 10 variable impact model. And we can see that it adds about 7 to maybe 11%, or increases the amount of variance by 7 to 11%, which coincides with what we saw in the previous slide. And then here in the blue is the added explanation of the full model compared to a model that only contains about 700 static variables. So they essentially just took the dynamic variables out. And so we can see that if we're thinking of this as the added comparison with the full model, it's about maybe 2 to 5% of an increase when we add those dynamic variables over time. And then here they also showed some examples when they stratified their data set by patients who had shorter ICU stays, so about 5.5 days, versus patients who've had longer ICU stays. And so here in the blue are the patients with the shorter ICU stay. And we can see that overall the explanation of variance is a little bit higher than in patients with longer hospital stays. And we don't really see this increase in the longer hospital stay right before discharge. So suggesting that this model may not be performing, or this modeling strategy might not be as useful for patients with longer hospital stays, which we could think of as likely being more severe and having a more complicated treatment course, which the model seems to be having difficulty capturing. And then next is kind of combining their entire modeling approach. And they provide this nice depiction of one individual patient's clinical trajectory. So this is an example of a patient who was 50 years old. They had a moderate TBI and they had blood in their brain, which required surgery. Their outcome at 6 months was a GOSE of 2 or 3, which is a vegetative or lower severe disability state. And so breaking down this quite busy figure, if we look here at the blue line, the first blue line, this is showing us the GOSE greater than 1. And so showing us what is the probability that this patient at least survives. And we can see in the first few days at the start of ICU admission, this probability is fairly low. But then we see it increases quite a bit where in the green box where it says this high magnitude transition. And so this is indicated as as this high magnitude transition point where the patient appears to be clinically improving. And then they use that time shop approach to look at which specific variables seem to be most influencing the model in the improved prediction at this point. And so they point out in their discussion that the administration of DVT prophylaxis seems to be associated with this improvement. And so they demonstrate this as a way to be able to identify specific medications or interventions that could be contributing to either improved or declining patient outcomes. And so this was kind of a fast overview, but in conclusion, the modeling strategy presented here allows for the identification of clinical events associated with important prognostic clinical transitions. I think some important takeaways are that simpler models can perform fairly well. Adding over 1,000 extra variables did not appear to substantially improve the predictive ability over the simplified 10 variable impact model. Additionally, it was interesting to see that only 91% of the model explanation was derived from the static versus the dynamic data. And they also concluded that modeling patients with longer hospital stays was a more difficult task, which I think for the next slide, I think brings some important future directions. So these patients with longer ICU stays are likely more severe and require more heterogeneous and complicated treatments. I think these are the patients we want to focus on, and we need more dedicated efforts to better understand this population and be able to leverage longitudinal data to better understand their clinical trajectories. I also think it would be interesting to see this approach adapted to a more acute outcome. So something like hematoma expansion, which is occurring within the first 24 hours. This could have higher clinical utility in order to help us understand which predictors or which interventions might enable improved, that might help us either predict hematoma expansion or be related to improving the patient's condition. And so, yeah, I'm excited. I'm happy to answer any questions and excited to discuss the paper a little further. Thank you for listening.
We do have one question. What data set was used for this study?
Yeah, so this is the Center TBI data set. There's a lot of information on it online, and maybe I can put the link in the chat, but it's a very, it looks like a very, we're trying to access the data set here at Northwestern, but it looks, it's a very organized data set, and there's been hundreds of publications that have used this data set. So it's very clean and has a lot of different directions that people can pursue in terms of research projects.
One other question was unrelated to the main point of the paper. Saya said she did see that the full-time employment was one of the most important variables associated with the score. Do you have thoughts on what that might be?
Yeah,
I'm not sure if I saw that right, but I think it was the one slide before that.
Yeah, the trajectory one.
Yes.
Yeah.
Can you go back 2 slides? Yeah.
Yeah, thanks, Saya. Let's see.
On the
full-time
left
employment.
bar graph and the bottom left one.
Yes, and I think that's also associated with some of their other results in the paper where if you were unemployed, you had poorer recovery. And so I'm not sure if I'm well equipped to answer exactly why that might be, but I think I would guess that it has to do with socioeconomic status. And so if maybe you're unemployed, you might be living in a poorer area, or you might have maybe comorbid conditions or disability that is preventing you from working. And so maybe that could be an indicator of just a patient's overall health status. I don't know if that makes sense or
Yeah, I think that
your
makes
question.
sense.
Yeah.
Yeah, I think that actually gets into the social determinants of the health space, like employment and also convenience to getting health care. Do you know if the paper actually systematically integrates some of the SDOH factors as the variables?
Yeah, so they don't really talk about that throughout the paper, but they do have a number of variables like employment status. I'd have to check if they have like, it's in Europe, but if they have some form of zip code or area, but they do contain quite a bit of the socioeconomic status variables.
Yeah,
So
I
I think
think
that's
this.
interesting.
Yeah, I think this might be a good direction for future research to this line of work and also to any clinical predictive modeling type of work because I mean just from our own group we find that by integrating SDH variables with those clinical physiological variables you would be able to not only improve on the accuracy, the overall accuracy, utility of the model, but also improve on the equity of the model. So this is something that we could potentially add in and achieve both goals at the same time.
There was
Mhm.
another response in the audience which said it could be correlation instead of causation. The person might be retired and older.
Yeah, I think that's a great point. And I think we should interpret all of this as correlations rather than causations. And yeah, so this type of variable, I think it's probably safe to say it's serving for a proxy for like older age or comorbidity status or socioeconomic status. And so, yeah, this is, definitely correlation versus causation, but can help us further understand which patients may be most at risk.
Great. I don't think we have any further questions, so we can go to the next one. All right, Saya.
Awesome. So hi everyone, my name is Saya. I'm a fifth year PhD student in Yikuan Li's lab, and I'm excited to present this paper to you guys today. Next slide, please. So I picked this paper titled Spatial Host-Microbiome Sequencing Reveals Niches in the Mouse Gut that was published in Nature Biotechnology in November of 2023. I think it would be a nice change of pace from Meghan's paper. This is a lot more focused on the technology itself as a new kind of method of biological inquiry. Next slide, please. All right, so I wanted to start with kind of like the big picture. And so this is a slide that I put here just to show that the microbiome as a topic has gained so much popularity in the last few years. And the reason behind it is that it seems to affect virtually every aspect of health. And I don't know if folks have read any popular science books that have been published in the last few years, but it's really become a really popular topic. And as you can see on the slide, it really affects anything from your neurological disorders, the metabolism, fetus development. We also have some things related to autoimmune diseases. And in the bottom right, I also added a paper studying the microbiome of the soil, which affects environmental issues and in turn our health as well. Next slide, please. All right, so a little bit of background related to this specific paper. I wanted to talk about the microbiome of the gut specifically. So the reason why the gut microbiome is important for our health is because it kind of lies at the surface between us and the outside world. So it can work in preventing pathogen colonization in our gut. And as we have seen in many other studies, it also mediates other host tissue functions, not only in the gut, but it affects other parts of our body through the local interactions in the gut. So sorry, go back to one slide, please. So in terms of what work has been done before and what remains, there has been many studies on mammalian gut spatial transcriptomics, so studying kind of how the transcriptome looks like in the gut of the host. But microbiome spatial mapping, there's more work to be done in that domain. Namely, most studies that does spatial mapping of the microbiome currently mostly focuses on family level taxonomy assignments. So the granularity of the taxonomy of the microbial population is relatively low. And lastly, metagenomic plot sampling, which is the current only approach for spatial bacterial sequencing in situ, does not profile the expression of the host. Next slide, please. All right, so here is kind of the overview of the technology that they are presenting in this paper. So all the way at the left side of the slide, we have kind of your normal spatial transcriptomic slide that you might see. So the authors take a very similar approach to this, but the mouse gut includes both the host tissue as well as the microbial population. So if we could focus our attention to the middle part of the slide, the authors developed this technology to profile both the microbiome and the host expression, and use the data that they obtain from that to study relationships between the host expression and the microbial signal. Next slide, please. All right, so to talk a little bit about the samples that were used to test out this brand new technology, the authors used germ-free mice, specific pathogen-free mice, and altered Chandler flora mice. So I am not an expert on these kinds of studies, but just kind of as a takeaway here, for all three of these categories, the authors basically know the microbial population. So they know what they are measuring to test out this technology. And the germ-free mice obviously has no microbial population, and the SPF and the ASF mice, they kind of know what population of the microbiome the mouse harbors. Next slide, please. All right, so in terms of how the authors went about profiling both the host expression, which is eukaryotic RNA, and the microbiome expression, or the taxonomy, which is bacterial, so very, very different types of transcripts. This slide is to explain like how they went about detecting both types of transcripts. So on the right-hand side, there's kind of a diagram of how they developed this hardware. So essentially, the hardware, the spatial transcriptomics slide is pretty similar to what you would use for normal spatial transcriptomics. But essentially, they incubate the slide with some probes that are specific to the microbial transcripts, so that roughly 50% of the spots have the 16S capture probe hybridized. So the 16S is a region of the bacterial genome that allows us to assign taxonomy to the microbial population. So essentially 50% of the probes on the slide can detect the microbe expression. And then the other 50% we can use for the host transcriptomics. Next slide, please. All right, and another kind of a novel element that the authors present in this paper is the taxonomy assignment for the microbial population. So Kraken-2, shown on the top left part of this slide, this is kind of the existing taxonomy assignment tool, and it was outstanding for its high speed and memory efficiency. So this is kind of software that is commonly used in the metagenomics research community, I guess. And what the authors did is on top of Kraken-2, they built a deep learning microbial taxonomy assignment model that utilizes both convolutional layers and LSTM layers on the transcripts that they observe. So the taxonomy assignment performance is shown on the top right part of the slide. So we can see that Kraken-2 performs really, really well, actually, all the way up to the family level of taxonomy. But Kraken-2 plus the deep learning model that the authors developed can perform well in terms of the classification at a higher granularity. Next slide, please. All right, so because this is a brand new technology that the authors developed, they really need to make sure that they are capturing the microbial population in a sensitive and specific way. So all the way at the left part of the slide, we are showing the bacterial transcript alignment rates. So here we see that the germ-free mice have pretty much 0% bacterial alignment, which means that we're not getting any non-specific signal. So this is good. And the authors also compared their signal from SHM-seq, which is their new technology, to FISH. So fluorescent hybridization signal of the spatial tissue. So the figure all the way at the right, there's a lot going on here, but basically what we need to see is the x-axis shows the SHM-seq signal, and the y-axis shows is the fish signal. So the takeaway here is that basically the microbial population are pretty well correlated between the two technologies. So this is kind of their way of saying that the signal that they're detecting from this new technology is sensitive, but also specific. Next slide, please. All right, another thing that is really important to know about the approach that they took on this paper is that the authors defined what they refer to as morphological ROIs or MROIs of the gut tissue. And they basically aggregate the signal coming from all the spots that belong to the same MRI. On the top half of the slide, they're showing kind of a diagram of what the MRIs look like and how that compares to the original tissue image on the left-hand side. The color coding is not very clear in this figure, unfortunately, but I think the really important point here to understand is that they use this MRI approach as opposed to treating each individual spot as an individual signal point. And on the bottom part of the slide, they're showing that certain genes are correlated pretty well between germ-free and SBF mice. So for each of the three kind of pairs, left side shows the germ-free, right side shows the SBF. And we see that the signals of these genes are pretty well correlated between the two. Next slide, please. Here's another slide showing, I think that it was really important for the authors to make sure that their technology is reproducibly showing good signal. So on the left panel, we are seeing signal of certain bacterial genera, and the different box plots show the various MROIs. So here the authors are showing that we see microbial genera or genus genera that we expect to see in certain ROIs. So this is showing that we are seeing expected signal in certain areas of the spatial area that we observed. And on the right-hand side, the authors are showing multiple tissue slides, their annotation and their bacterial signal to show that there is robustness and agreement between multiple slides, tissue slides. All right, next slide, please. And of course, for biological discovery, they had to look at genes that are differentially expressed based on the presence or absence of the certain microbial populations. So here, the blue colors in general are showing the bacterial abundance, and the red color is showing the host gene expression. And we can see that certain microbial genera are only observed when certain genes are expressed in the host tissue. So I think there is a lot more that can be done with this technology, but maybe we can move on to the next slide now. Yeah, next slide, please. Okay, I think this is a nice segue into the limitations of SHM-seq. So the biggest thing is the low spatial resolution at this point. And this was limited by the resolution of the solid phase capture arrays. But the authors tried to get around this issue by the manual annotation of each spot, assigning each spot into an MRI and aggregating the data from those spots to achieve robust signal from each kind of tissue type. So this gave us some nice interpretations in terms of the biological question that the authors were interested in, but there's a lot more that can be done with a higher resolution slide. So the authors leave this high resolution work to future studies. And another thing, another limitation that was mentioned in a letter associated with this article was the difficult application in vivo. So because of the invasive nature, obviously, of the tissue sampling, it limits the technology, the applicability of this technology in vivo. But it was also mentioned that integration with some emerging non-invasive sampling technologies might provide some further avenues to overcome these limitations. So yeah, I thought this was a very fun paper in terms of the new technology in relation to the gut microbiome being kind of a field topic of research that is gaining momentum. So yeah. I'm not sure if there's any questions.
We don't have any questions, Saya. Great.
Okay.
Thank you.
Sounds good.
If there are any questions as we go through, please enter them in the Q&A and we can make sure to address them.
Absolutely.
Thanks, everyone.
Shall we move on to the next presentation? Yikuan, you can take it away.
Right, thank you. Hi everyone, my name is Yikuan and thank you so much for joining us today. So I'm gonna present this paper that was recently accepted by ICLR 2023 as deep reinforcement learning for cost-effective medical diagnosis. It's in collaboration with our labs and some precedent folks. Can I go next? So a little bit of background about reinforcement learning. So unlike some other area of machine learning, let's say deep learning, NLP, reinforcement learning is largely explored in the healthcare domain. So I'm gonna give a little bit background on reinforcement learning. So it's an area of machine learning concerned with how intelligent agents can take actions in the environment in order to maximize the notion of cumulative reward. And so basically reinforcement learning is all about sequential decision making. And some notable application of reinforcement learning is for some Pac-Man agent, like an agent that can play Pac-Man games. And the AlphaGo, the Go game player back-ended by reinforcement learning can defeat all humans and the recommendation system that we are using every day. And can I go next? Okay, so so think about how do we formulate a machine learning project, let's say a deep learning project. We should have it's pretty straightforward. We have an input, probably high dimensional features, and we have the outcome. So probably a binary outcome if the patient is at risk or not. And after we having the input of high dimensional features and outcome, so we can feed in some deep learning models, let's say transformer, CNN, etc. So we can start the training process. So similarly for RL, we should have a setup before we use the deep learning models, and it's called Markov decision process. It can be used to model the sequential decision making process. And it has 5 elements. I'm gonna use an example of the Pac-Man game here. So the first element is S is size of states. If for this case, it's probably the coordinate of the Pac-Man. And the size of actions is come to the next element is for this case, it's the, excuse me, the Pac-Man can go to the right or to the left. Those are 2 possible actions at this state. And the third one is the transition function that after taking certain actions, so what's the next state based on the current state? Let's say if a right is issued by the gamer, so the dot move to the right coordinates. Sorry, can we go back? Okay. Yeah, here we go. Yeah, and next is the reward function. And so it's the immediate reward received after transition from the current state to the next state. For some cases, if a Pac-Man eats a dot, so it's gonna be a small reward for the Pac-Man. But if it's got caught by the ghost, it's gonna be a penalty. And if finally this Pac-Man escapes from this puzzle, it will be a huge reward in the final type step. And gamma is the discount factor that controls the future rewards, and it's usually a hyperparameter that we can choose from, probably some value approach to 1, let's say 0.99. And so after this setup, we have the formulation of a study, and we can RL seeks to find the optimal policy to construct a trajectory that has a maximum cumulative expected reward, also called the rater. Can I go to the next slide? Yeah, so this is one of the most remarkable and groundbreaking study that using reinforcement learning in the healthcare domain. So it's about sepsis treatment. So the idea is that the authors try to seek improvement on the doctor's treatment strategy of using different dosage of IV fluid and vasopressor to treat sepsis patients. And those, so the Markov decision process for the study is that the site of stasis patient's conditions at every 4 hours. So the data is from ICU extracted from mimic 3, I think, and they quantize the patient's status for every 4 hours. And sites of actions are the dosage of IV fluid and vasopressor, and they quantize the dosage of IV fluid and vasopressor both to 5 different levels, and in the combination they have 25 different actions to select from. And transition functions like after this action or medication has been given, so what's the patient's next conditions from also drawn from the data set. And the rewards is based on the intermediate rewards, except the final step right before the patient was discharged, is that the change of SOFA score. If there's an increase of SOFA score, there's going to be a penalty. If it's a decrease in SOFA score, that's going to be a reward because patients are getting better. But for the final step, if the patient discharged alive, it's going to be a reward. Otherwise, if patient is deceased, it's going to be a huge penalty. And they use the discount factor 0.99 here to control the future rewards. And after signing this up, they use the deep learning models, and they have a new dosage recommendation to treat sepsis based on some off-policy evaluation check, because the patients are drawn from the behavior of the doctors, and however, they propose a new treatment strategy, and they have to go through some off-policy evaluation check. There are a huge mathematical proof and guarantee behind this, but they can evaluate the call that the new model can be better or comparable to the doctor's strategies as of prescribing IV fluid and vasopressor. Can I go to the next slide? I see there's a question in the chat. I'm not sure what difference between state and the reward here. And if we go back to one slide before, so the state is the patient's condition. They all have many different information, probably patient's medication, medication history, and demographics and other lab measurements. But the reward is scatter, just one number to describe if the SOFA score is increased or decreased. So I hope that will answer your question. Okay. And so let's go back to this study. So what's the motivation and the objective? So we try to investigate the use of reinforcement learning for lab test panel optimization. So when we are developing machine learning problems, and we found that sometimes there are data missing. Let's say we want to predict an outcome based on the lab measurements, and there are some missing in the data. And with some imputation methods, we found out that the model performance is still comparable, even comparable with the model developed using full observations. So this reminds us that sometimes that we can make early diagnosis if some test results are not right, or if we just don't even give this test to the patient, we can still make a comparable diagnosis in terms of accuracy. And by doing this, we can save the cost for patients. So we call it cost-effective diagnosis, because we want to use minimum lab tests to maintain a high accuracy of diagnosis. And so our new tests get new results and give a feedback on the diagnosis. It's a sequential decision-making process. So it's pretty natural that we can use reinforcement learning. And can we go to next? Okay, so here's the whole setup or formulation Markov decision process for the study. So at the first time step, we only have the patient was just admitted to the hospital or inpatient. We only have demographics information, vital signs, and medication history. And from there, we start trying to recommend the new test for a patient. We first have an imputer to impute the existing information to the latent space as a hidden state. And with this hidden state, we ask the deep reinforcement learning model. So they have a some action to select from. So they can select to order new tests for a patient, or they are confident enough to make a diagnosis. For the first time, the model recommends to take the test number 2 and test number 4, and it incurs $48 for these 2 tests, and it goes to the next time step. So we have the results from test 2 and test 4, and we still using the same imputer to map it to the lattice space with the hidden states. And we repeat the process as the deep reinforcement model. So what actions do you recommend? And they recommend 2 more tests. Test number 1 and number 3 incurs the cost of $194 and move to the next stage. And we keep repeating this process until the model, at the final step, the model thinks, okay, so they are confident enough to make a final diagnosis, and we call the classifier to kick in to make the final diagnosis. And this is the whole process, iterating process of how we make decision, how we do the sequential decision making. And the state of each time step is the imputed hidden states from the existing information with an imputer. And the reinforcement learning model recommends the different actions. And the action can be selected from either to select more tests or do the final diagnosis. And the reward is just the cost, like how many dollars to be spent on those those tests. And we also, the transition function is that after the tests are ordered, we have the new results and move to the next step. And the discount factor, we also use a pretty common discount factor of 0.99. Can we go to the next step? So a little bit technical details of this setup. There are 3 kernel modules for the whole model. The first is the imputation module, mapping from the existing information to the latent space of hidden states. And we use an existing model called EM-Flow for as an imputer. And for recommendation module, the deep reinforcement learning model, we use the PPO. It's a pretty common deep reinforcement learning model developed by OpenAI. And for the classification module, we use the multi-layer perception or fully connected. Can we go to the next? So here comes the results. We experiment our study into 3 different data sets, and the outcome is different. The first one is the abnormal in ferritin, and the second one is the diagnosis of acute kidney injury, and third one is the mortality for sepsis patients. Those are the final outcome. And we've compiled our models. Our models are in the last 2 rows. They have a slightly different setup with all the baseline models. We can see from lossless regression, random forest, and etc. They use all the test panels available to make the final diagnosis. So we evaluate the model based on two different perspectives. The first one is how accurate the model is. We use the F1 measure and the AUC to evaluate. And second one is what's the cost associated with the diagnosis. So for the baseline models, they use the different all the test panels. They can, let's say the ferritin data set, they have an average cost of $290 for a patient, but our model only requires $62, and the performance is competitive to those models using full observations. So our F1 is 0.624, is just comparable to 0.627 with the models using all the observation. And we have the similar observation with 2 other data sets. So in both AK and sepsis data set, our model just probably 25% of, 15% of the whole cost. So we probably have $90, and the full observation requires around $600. But our IFM measure and AUC are comparable or even better than the models using full observation. So we can count that with our reinforcement learning model and setup, we can achieve comparable performance to the model, those using all the lab tests that are available, but we save the cost for the patients by a lot. And can I go to the next slide? So by reshaping the reward function, we can adjust the balance between the cost and the diagnosis accuracy. So here's the parental fronts for maximizing F1 measure on those 3 data sets. And the y-axis are F1 measure or AOC, but the x-axis are testing costs. As we can see, as the increasing of the testing costs, there is the increase in the FM measure or AURC until a certain turning point that even though we have the more testing costs for patients, the FM measures are still stated the same. So we can have this parent front and to adjust the trade-off between the testing costs and the FM measure. And at those turning points, we have the minimum testing costs, but achieve the highest FN score. And can I go to the next slide? So the future direction of this study, so right now we only consider it's more like a proof of concept study. We can recommend the test panels at certain time stamp. We can now sequentially recommend more tests, lab test panels. So we want to incorporate another temporal horizon. And also we want to involve additional modality of tests. Right now, all of the test panels are structured data, but we want to involve more tests, different modality of tests. Let's say image, they are slightly different. And so this paper has been published as a conference paper at the SALR 2023, and here's the link of this paper. Yeah, that's pretty much for my presentation. Any questions?
There are no questions in the Q&A. Yuan, did you have any additional things to add?
Yes, so I think these are great presentations. So thank you, Meghan, Saya, and Yikuan. We had a very nice composition of the state-of-the-art works from both the bioinformatics side and the clinical side. We will also love to ask the audience for volunteers for presenting some of the work that you think will be great and cool to share to the group and to the not only people at the Feinberg School, but also at the health system so that we can exchange ideas. Also, if you have something that you would want to know, but you don't feel confident in presenting, please also let us know so that we can ask people that might be familiar with that areas to do a quick intro so that people can get the takeaway of what's the intuition and how they may benefit your own work. And in either cases, please do let us know, and then we will coordinate those into our future sessions.